So this feedback video is going to look at the first required practical, which was an effect of a named variable on the rate of an enzyme controlled reaction. In this one, the named variable was temperature, so temperature was the independent variable, and we were measuring the rate of reaction. As you'll see later on, you do this by measuring the time taken for the cross to appear. To get a rate from that, you have to do 1 divided by time. So our dependent variable could be time taken for the cross to appear, which we will then use to calculate the rate. With any method, you need to make sure you keep your control variables the same. This is stated within the method and within the list of ingredients. So here we are keeping the concentration of enzyme the same, keeping the concentration of substrate the same, we're keeping the pH the same using a buffer, and you make sure it's at the correct temperatures using your large beaker to use as a water bath. Remember, it's important that you only want temperature. So you only want temperature to affect the rate of reaction. That is why you need to keep these three the same, because they would affect the rate of reaction. If it was not kept the same, the results would not be comparable. Other control variables are sort of written in the method points, so it states the volume of enzyme that you would use, it states the volume of your substrate that you will use each time. The reason you need to keep the tubes in the water bath, you need to keep the enzyme and the substrate separate for 10 minutes to allow equilibrium to reach before you mix them. Again, it would not be comparable, your results, if they were not at the correct temperature that they should be before you mix the enzyme and substrate. One huge limitation with this practical is point 9, how you actually measure the time taken for the cross to it's very subjective. Okay, very subjective judgment. Do you just see until you can see the tips of the cross? Is it until you can see the whole of the cross? Also, each person's eyesight is different, so it's a very subjective judgment. So it makes not a very sort of a very accurate method of measuring. Do the experiment three times, so you get 3 repeats, and you can calculate a mean. Really, what you should do with the 3 repeats is check that they're concordant, so they should all be similar. If they're not, really, you should be doing another repeat, so you can then identify which results are the anomalies and eliminate them from your mean. So that makes the results much more reliable. It makes the mean much more reliable. By process data, what that means is you've got to do something with the data you've collected. So at the very least, calculate a mean. And we're actually going to, what this means in this case, is to draw a graph. So your graph should be rate of reaction up the side on the y, which should be calculated by 1 divided by time, and temperature, your independent variable, on the x. So over the next few pages, I'm just going to go through some of the questions, not all of them. The mark scheme is at the end of this presentation. So not all of them, just the key questions that are quite difficult to understand from the mark scheme. So the first one is a calculation. It asks you to calculate the rate of reaction between 0 and 2 minutes. So first of all, make sure you're using the correct times. And then you need to go up to the line and across from each of those. So 0, 0, that's fine, that's touching at 0. At 2, go up to the line and across. You find a value of 0.8. A rate of reaction is calculated by the change in y divided by the change in x, where x is time. So 0.8, the volume's gone up by 0.8, divided by the time, which is 2. So it's 0.4 per minute. For this question here, yes, as you can see, the rate of reaction is slower at 20 degrees because it's less steep. If you allowed it to continue, eventually it would reach the same volume of product produced as the higher temperature. It would take longer to reach that point because there's less kinetic energy. So it would eventually reach the same volume of product. So that's when all of the substrate has been converted into product. That is when the graph would level off. For this question here, you can see the line has leveled off at that point, so there is 0. For this question here, we've got between 80 and 90, not a very steep curve at all. Between 10 and 20, we've got a steep curve. So the rate of reaction is much faster here than it is here. So it's slower here because you should know that as the reaction proceeds, the substrate's used up. So if the substrate concentration is less here, there'll be fewer collisions with the active site of the enzyme, so there'll be fewer enzyme substrate complexes. The blue line here indicates the next question, where it's asking for what will happen to the rate of reaction as time increases. So at the beginning, the steepest part of the curve here, which is when the rate of reaction is fastest, there's the highest concentration of substrate. That means you've got the highest rate of reaction, so using this scale here. So the highest rate of reaction here. As time proceeds, the rate of reaction gets less and less and less, and kind of mirrors this curve as the substrate concentration decreases over time. Fewer collisions, so the rate will decrease as time increases. Eventually you have a rate of 0 as there is no more volume of oxygen being produced, no more product being produced. For question 8 part B, so not this question here, the one on the next page, but I need to use a diagram. We've got here which is a tangent to the curve, so the initial rate of reaction from 0, 0, what's the angle of the curve, and follow it with a straight line. Now we need to use that to be able to calculate the rate of reaction, because as this line here is curved, you can't accurately work out the change in y divided by the change in x, because it's not the same values going up each time, it's not proportional. So we have to draw a tangent to the curve to calculate the rate. So exactly the same way as if it was a straight line. So the difference in y divided by the difference in x. So the difference in your concentration divided by the difference in time. Choose any points along the line where it meets. So you could go across from 6 to there and then down to a value of about 5. So you do 6 divided by 5 to give you your rate of reaction in arbitrary units per minute. Question 8c seems to be a bit of a strange question, but all you need to do is just follow the equation really. So when t is 15, find what the rate is. Okay, so for t is 15, the rate there is 4. So the rate at t would be 4. The rate at t plus 10, so that would be 15 plus 10, so that's 25. So the rate at 25 is a value of 8. So 8 divided by 4 gives you an answer of 2. As we have not done respiration or active transport yet, this question won't make that much sense at this point, but later in the year that will. The key to this is that if temperature is having such a dramatic rate on the uptake of potassium ions, then it must be involving enzymes. And the reason it involves enzymes is because active transport requires ATP, and to make ATP you need a process of respiration, which involves enzymes. You can't carry out respiration without enzymes.